Hello everybody. Today's topic is recombinant DNA technology. It is also called as genetic engineering or cloning, and short form is used as RDT. So before starting with recombinant DNA technology, let's just brush up our knowledge on basic terms. DNA. What is DNA? DNA is the molecule which has all the genetic information in it for carrying out all the functions in all living organisms. And the DNA molecule is made up of nucleotides. So repeated units of nucleotides, they make a DNA molecule, and it is present in a double helical shape, a double stranded DNA in which nucleotides are joined together to make a long polymer chain. So this is how it looks like. And coming to the next term, gene. Gene is the stretch of DNA. Now this stretch of DNA has different sequence of nucleotides attached to one another. The sequence of nucleotides will be different in different genes. So this is a stretch of DNA which is capable of determining a characteristic. Now this characteristic could be anything: your height, your eyes color, your skin color, anything in your body. Any characteristic in your body is due to a particular kind of gene or a particular sequence of DNA which has that information for that particular characteristic. So next is replicate. It means making exact copies of something or duplicate something. Then is isolate. Isolate means to separate something, to get something from a bigger product, just separate it from there. And another very important term that we have is cloning. Cloning also means making copies, making exact copies of something. It could be DNA, making copies of DNA, making copies of genes, making copies of cells, making copies of a full organism. So making exact copies or making any duplicate of something. The next term is vector. Vector is a DNA molecule which serves as a vehicle to carry the gene of our interest to another living cell where it can replicate So vector is the vehicle in which we can insert our gene of interest, and after insertion it can take it to the another living cell where it can replicate. Then we have amplification. It means massive multiplication of something or massive duplication. So these are the important terms to be used in recombinant DNA technology. So let's start with the basics of recombinant DNA technology. Recombinant DNA technology is based on gene. Gene is a stretch of DNA which can give you a characteristic. So the first thing we need here is the identification of gene. Which gene we want to study, or which gene we want for our work. So we have to identify that gene. Suppose we have to identify any human gene. Suppose we have to identify insulin gene, the gene for producing insulin. So in humans we have 3 billion base pairs of DNA. So from such a big DNA or genome of human, it is very difficult to identify the gene for insulin. So there are different methods to do that, but this is just an overview. I just want to give you the basic knowledge. So here let's just stick to the plan. First thing that we need is to identify the gene. Then the second would be to isolate that gene, to separate that gene from that big genome. So that means we have to separate the gene which makes insulin from the human genome. So there are different methods. We have to make cuts in the human genome, and there are enzymes called as restriction enzymes. These enzymes are used to cut and isolate the gene of our interest. So the third one would be, after isolation we have to amplify or multiply the gene so that we can study it or any other purpose that we need that gene for. So we have to amplify or multiply. So there are different methods to multiply or amplify the gene. The one most commonly used method is by cloning, and it goes like this. Suppose we have isolated a gene of our interest, and we need a vector. This is the vector. Vector, if you remember, is the vehicle in which we can insert our gene, and this will take our gene to another living cell where it will be replicated. So the gene of our interest is to be inserted in the vector first. So this is how we have inserted our gene in the vector. Now there are different kinds of vectors available. The most commonly used vector is plasmid. This is extra chromosomal DNA from bacteria. So this is the vector which is used commonly. So after insertion of our gene, we get the product which is called as recombinant DNA. The word itself is telling the meaning. Recombinant means recombine. We have taken DNA molecules from different sources. Vector is also a DNA molecule from bacteria, and gene is also a DNA molecule from different source, but this gene is of our interest. So we have rejoined the two DNAs from different sources, and the product that we got is recombinant DNA. This is recombinant DNA. Once we have made this recombinant DNA, the next step would be to introduce or to transform this recombinant DNA to bacteria. As we have taken a vector called plasmid from bacteria, plasmid is capable of replicating in bacteria. So we will introduce this recombinant DNA that we have made to bacteria. So transformation is another word for introducing the rDNA, our recombinant DNA, to bacteria. So when we introduce it, it would look like this is the genome of bacteria, and this is our recombinant DNA that we have made. So this is called as transformation of recombinant DNA. This is transformed bacteria. So now we have to find the way by which we can pick the bacteria which is transformed. Find the transformed bacteria. Please note there are different ways to transform the bacteria. There are different ways to introduce our recombinant DNA to bacteria. I will be covering all those details in upcoming videos. This is just a brief overview of recombinant DNA technology. So we find the transformed bacteria. Once we find the transformed bacteria, we will grow it in the medium. Medium is a nutrition that we would provide to this bacteria so that it can grow, it can multiply. So when this bacteria will multiply, suppose we have this medium provided. So these are the bacteria which are multiplying here. So when we picked only the transformed bacteria, sorry. So when we picked only the transformed bacteria and we have given the medium to multiply, so these will be the exact copies. These bacteria will be the exact copies of each other. So we can also say that these bacteria are the clones. They are the clones of each other. It's a clone. So in this, the gene of our interest also replicated, and the bacteria is also replicated. So we have two kinds of clones here. One is the bacterial cell clone, another is our gene that we introduced. So this is how we have amplified, we have multiplied the gene of our interest. Suppose we took the insulin gene. So when we have multiplied this insulin gene by this method, now we have the multiple copies of that gene which can produce insulin. So more insulin can be produced now, and that can be used for different purposes. There are many ways in which we can use that amplified production of insulin. So this is how it works. So I would love to see the feedback on the comment box, and also if you want me to cover any other topic, please leave your message on the comment box. I will make another video with your topic, and have a nice day. Bye bye.